Part 2 of our DNA replication videos. In part 1 we talked about the DNA template strand, and it runs from 5' to 3', and that's the 5-carbon sugar being on top, and then the 3-carbon sugar pointing downward on this side, and then the opposite strand runs from 3' to 5'. And so what we call this is anti-parallel, meaning that they're running side by side, but in opposite directions. Then we talked about DNA helicase, which is an enzyme really important in DNA replication, because what it does is it unwinds our DNA helix to make way for other enzymes to get in here to copy the DNA strand and to put new nucleotides in. We also have topoisomerase is another important enzyme that what it does is it relieves the stress from the DNA helicase, unwinding the DNA. And then we also talked about these single-stranded binding proteins that latch on right here at the replication fork to prevent these complementary base pairs from rebonding to each other while replication is happening. Okay, so the next enzyme we're going to talk about is RNA primase. So what this does is it builds on short complementary strands of RNA on the template strand. Alright, so we're going to write that. Builds short complementary segments of RNA. So RNA, remember, is ribonucleic acid, onto the template strand. And these primers, what these primers do is they provide a starting point for DNA polymerase 3 to work from. And we're going to talk about that. But basically they're like, here, this is the start of our sequence, now you continue from here. So these primers provide a starting point for DNA polymerase 3 to build from. So DNA polymerase 3 is also an enzyme. Alright, so I'm going to color my RNA primase pink. Right here, pink. And then I'm going to find anything labeled number 4. So I have a number 4 on this side. And I have a number 4 down here on the bottom. Right here. Alright. Next we're going to move on to that DNA polymerase 3, number 5. So this is a really important enzyme. These are all really important enzymes, because it adds nucleotides onto the end of the RNA primers. So adds nucleotides onto the end of the RNA primers. Okay. On the leading strand is the strand that goes from 5' to 3', and in this case is on top. The leading strand, only one DNA polymerase 3 needed, because strand is synthesized or created continuously. So when we say continuously, we mean in one long section, all the way down. On the lagging strand, we need many of these DNA polymerase 3 because we are creating fragments as we go. Fragment here, and we call those Okazaki fragments. Because Okazaki fragments are made. So these are those Okazaki fragments. They're called Okazaki because this husband and wife team with the last name Okazaki created or found them, discovered them. Okay, so I'm going to color my DNA polymerase 3 orange. Orange. Okay, and my orange right here, DNA polymerase 3. Adding on to what the RNA primase has already started. I also have a DNA polymerase 3 right here. Here, my apologies, and also right here. Okay, next we're going to move on to the nucleoside triphosphates. Basically, these are like normal nucleotides. Very similar to normal nucleotides, except for they have extra phosphates. Extra phosphates. These extra phosphates give energy for DNA to be synthesized or created. I'm going to say synthesized. We're going to stick with the word synthesized. Whew. Or we can write that synthesized. Okay. So I'm going to color my nucleoside triphosphates in brown. Alright, and so actually first let's put our primers in. So our primers are right here. That's what those RNA primase laid down. RNA primers laid down by the RNA primase up here. Also, we have them in multiple locations here, right here, right here. We have them being swapped out right here. A few more right here. Okay. And then my nucleoside triphosphates, I'm going to color brown. Brown. And so all of these are those nucleoside triphosphates. We have a few right here. Some down here. Being swapped in right here, swapped out right here, and then on the end here we have some more. Alright, right there. Okay, so those are our nucleoside triphosphates, number 6. Okay, number 7 is just talking about our leading strand. Alright, our leading strand is the strand that is synthesized from 5' to 3' based on the new strand. Okay, so this is 5', this way is 3'. This is our leading strand. It's now synthesized going from 5' all the way down. So you can see that there's a 3' up here, 5' down here. They're opposites because these are now like the anti-parallel strands. On this side, these were the anti-parallel strands. Now we have new anti-parallel strands being created. So the leading strand is newly created DNA. It's a newly created DNA strand built, in the same direction, built towards the replication fork. Towards the replication fork. Okay, it's built continuously. and requires only one primer. For RNA primase, only one of each of these things. The DNA polymerase, 3. We call that DNA polymerase. 3 DNA polymerase 1 and 1 DNA ligase. All things we'll talk about in just a minute. Our lagging strand is new DNA strand built away from the replication fork. Built in fragments. In those Okazaki fragments. And because DNA polymerase 3, it can only add DNA from the 3' to 5' direction of the template. So this is the 3' to 5' direction of the template, and so it has to be built in fragments because of that. So basically, there's like this fork here that's slowly unzipping, and we only have like so much space. Like we have to go in segments because like on this one, we're going in this direction, like kind of following what the helicase is doing. We're following what the helicase is doing. On the lagging strand, we're like working ahead of what the helicase is doing. We're like, okay, the helicase has done this much, let's go. Okay, now the helicase did a little bit more, let's go here. Helicase did a little bit more, let's go here. So this is built in segments. DNA polymerase 3 can only add in the 3' to 5' direction of the template. Which is the original. This means moving away from the replication fork. Okay. Well, let's leave that there for a second. Okay, number 9 is our DNA polymerase 1. And what this is it's the enzyme. It's another enzyme. So anything ending in ASE is an enzyme. And it removes all of the RNA nucleotides from the primers. So for example, like this here, removes all the nucleotides from the primers, and it replaces them with the appropriate DNA molecules. Okay, so I'm going to color my DNA polymerase 1 dark green. Right here, dark green. And then I'm going to bring that in here for our DNA polymerase 1, dark green. So I can see it's switching out what used to be primers and is putting in nucleotides now. So it used to be blue, it's swapping out and bringing in a brown one. So swapping out the primers for the nucleotides. And we just have one of those on here. Okay, number 10 is DNA ligase. And what this does is, this is the enzyme that basically joins all unattached DNA together. It forms bond between them. It's like the glue that holds your DNA together. DNA that joins unattached DNA by forming bonds between them, between sections. And so this is necessary for the Okazaki fragments coming together especially. Okay, so that's number 10. I'm going to color that yellow. So you see here, it's bringing those two fragments together. There's a joint, little joint there, bringing those together. Okay, last thing, exonucleases. And this is basically a way to help us edit our DNA. So errors occur in the DNA replication process. And these exonucleases, they're part of the DNA polymerases. And these allow them to these exonucleases allow them to remove mismatched DNA nucleotides and replace them with the correct ones. So the DNA nucleotides are replaced with the correct ones. Alright. So just pointing out here the major difference between DNA polymerase 3 and DNA polymerase 1. DNA polymerase 3 adds on from the primer on. The DNA polymerase 1 actually comes in and replaces these primers with nucleotides. I'm going to pause the video here, and then I'm going to make one last video that is kind of a recap of what we have all just covered, but you should have all of these boxes filled in, colored, a almost finished product by now.